Hello everyone and welcome to today's webinar titled Optimizing Transfection to Generate High-Titer AAV and Lentiviral Vectors. This webinar is part of the bioprocessing virtual event. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and Mirus. For more information about Mirus, go to mirusbio.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on that Ask a Question box located on the far left of the screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Tseng, a technical support scientist. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Sandy, welcome to you. You may now begin your presentation.
Hello, thanks for coming to this webinar. My name is Sandy. I'm a technical support scientist at Mirus Bio, and today I'll be discussing ways to improve AAV and lentivirus production through optimization of transfection. First, I want to give a little background on Mirus Bio for those of you who aren't familiar with us. I also want to touch on how these viral vectors, AAV, lentivirus, how they're being used for cell and gene therapies. Then I'll focus on how at Mirus we've examined ways to maximize on AAV and lentiviral titers through optimizing parameters that surround transfection. And I'll be touching on these parameters here, one after the other. And I'll end with a Q&A if you're watching this live. Mirus Bio was established in 1995 from the research of scientists at the University of Wisconsin-Madison. At the time, they were exploring the cutting edge of gene delivery, and they came at it from a very interdisciplinary approach that involved chemistry, physics, cell and molecular biology. And since then, Mirus has been known as the transfection experts. Here's a picture of some of us in 2019. That's me over here. To this day, we're still headquartered in beautiful Madison, Wisconsin, USA. However, our reach is international and we offer distribution and support for scientists around the world. Here, I just want to highlight a few milestones in Mirus history and how we've evolved over the years to meet the needs of the scientific community. Our first transfection reagent that is still a big hit today is TransIT-LT1. It was the first to be compatible with transfection of cells that were still growing in serum-containing media, and it did so with high efficiency and low toxicity, hence LT for low toxicity. And not a transfection reagent per se, but we developed a chemistry, label it, for tracking transfection through labeling of nucleic acids with tags such as fluorophores, and it did so without impeding gene expression of the nucleic acid. We also developed reagents specifically for high-efficiency delivery of small oligos such as siRNA, as well as larger RNA like mRNA with our TransIT mRNA kit. And we've also developed products for electroporation of cells that are difficult to transfect with chemical reagents. And TransIT 2020 and X2 are really next-generation transfection reagents. They were designed to transfect cell types that were previously untransfectable with chemical transfection reagents. TransIT X2 can also deliver Cas9 ribonuclear protein complexes. And to support protein production in suspension cells such as CHO and 293, we introduced TransIT Pro and our CHOGRO family of products. Now, and the topic of this talk today, is our recent developments with reagents for virus production. Scientists have been using our TransIT reagents for virus production for decades, but we noticed an increasing demand for virus in light of advances in gene and cell therapies coming to market. So to address this, Mirus has developed VirusGEN Transfection Reagents. Alright, so before jumping into the main part of my talk about optimizing transfection for virus production, I just want to highlight what I mean by advances in gene and cell therapies that use virus. So viruses get a bad rap these days, and obviously there's a good reason for that, but a non-pathogenic recombinant virus can be produced to house therapies for delivery using that same infectious potential. So what's the difference between gene and cell therapies? Well, what it comes down to is what form the therapeutic takes. So in gene therapy, the packaged gene is therapeutic, while in cell therapy, the engineered cells are therapeutic. And because the gene is the therapy, in gene therapy, the virus containing the packaged gene is directly introduced to patients, such as in the administration of AAV. In cell therapies, the viral vector is introduced to cells that have been removed from the patient ex vivo. The cells are then reintroduced into the patient, and engineering of patient cells is often performed with lentivirus or lentiviral vectors. So really right about the time Mirus was founded, the first gene therapy was licensed by the FDA and subsequently discontinued. That's what those pink colored boxes indicate. And not for safety reasons in either case. In the case of vitravine, the disease that it treated was being stamped out in other ways. Anyhow, this review from 2020 summarizes the 22 gene and cell therapies that have been approved for use around the world, which might not seem like a lot, but what is significant is the growth since the 90s accelerating to today. For Glybera, this was known as the $1 million drug because it literally cost $1 million USD per patient, which is obviously quite costly. And it was discontinued because it was costly relative to its utilization. And Glybera was an AAV-based gene therapy, and virus-based therapies, as you can see here in this pie chart, make up a significant portion of the 22 approved gene and cell therapies. We are likely going to see an increase in the number of approved gene and cell therapies as evidence of this. As of 2021, when this industry report was released, 1,220 active clinical trials involving cell and gene therapies are ongoing in these various phases. And this chart here shows the number of new clinical studies posted each year since 2000, which mention the use of AAV and lentivirus specifically. And as you can see, it's clearly growing. In order to support these therapies, that means a high quantity or high titer of virus will be required. And it's estimated that the dosage is around the order of 10 to the power of 8 or to the power of 10 in the case of cell therapies. And to remind you, patient cells are transduced ex vivo, and once edited, the cells can then be further propagated. The average dose for AAV-based gene therapies is larger. It's around the order of 10 to the power of 11 up to 16, depending on where and how much virus will be required for the treatment. And it's much higher because the vector is administered directly to the patient for transduction of affected tissues, and the virus may have to contend with clearance from the body. So it goes without saying, to maximize production of these vectors is desirable to support these therapies and make them affordable at scale. And the transfection step of AAV and lentivirus manufacture is a critical point to achieve this. So to remind you, AAV and lentivirus is often produced through transient transfection of 293 cells, which is step 1 here in these diagrams. And the AAV and lentivirus is collected post-transfection and is further processed. Okay, so on to the meat of the talk, which is to discuss the ways we can improve yield of AAV and lentivirus through optimizing this critical step in production, transfection. So before we get to the Q&A, we will discuss these parameters. Okay, so first, the reagent. TransIT-VirusGEN Reagent was developed by Mirus with this workflow in mind. We first screened our vast library of molecules and compounds to identify reagents that would result in the highest yields of AAV and lentivirus. We used a reporter GFP system where the AAV2 or lentivirus contained a GFP transgene. Secondly, we also wanted to ensure that our reagent would be compatible for both suspension and adherent HEC293 cells in either serum-containing or serum-free conditions. And another important parameter was that the protocol for use of our reagent would be robust and scalable, because we know how important consistency is to any process in addition to getting a really great titer. And so a couple of years ago, in about 2017, we introduced our TransIT-VirusGEN reagent, which accomplished all of that. And specifically, to address the needs of our preclinical and commercial manufacturing customers, we launched our select format of the reagent. This is going to be closely followed by the launch this year of a completely GMP manufactured version. What's important to note is that all of these formats, RUO, Select, and GMP, are identical in formulation. So any development work done with the TransIT-VirusGEN RUO reagent can be applied seamlessly to the use of the Select and GMP TransIT-VirusGEN reagent. And together with the launch of the GMP reagent will be the launch of our VirusGEN AAV Transfection Kit and VirusGEN Lentivirus Transfection Kit, or LV Transfection Kit. These kits include enhancer solutions which further boost virus production. These kits will also be available in RUO, Select, and GMP formats later this year. Okay, so we benchmarked our reagents against other commonly used transfection reagents that are PEI-based. So what I'm showing you here are functional titers for AAV on the left and lentivirus on the right in suspension 293 culture. And as you can see, the addition of the enhancer component of the VirusGEN kit greatly improves the yield of AAV compared to competing reagents, and the same case for lentivirus. So which reagent that you use obviously has a very significant impact on vector yields, but so can optimizing the cell culture conditions of your 293 culture. In this experiment here, we examined cell density at the time of transfection for XP293 cells used for the production of AAV. And we looked at using either the reagent alone or in the presence of the enhancer component, shown here in red. So as you can see, the cell density at the time of transfection clearly impacts the resultant titers, especially for when the enhancer component of the VirusGEN AAV kit is used. And for this particular system, we would recommend a cell density at transfection of about 3 to 4 million cells per mil. Optimizing cell density also applies for lentivirus production, and in this experiment, lentivirus titers were highest when cells were at 4 million cells per mil at the time of transfection. Shown here is data for an optimization experiment that we performed for adherent cells. They were T17s grown in serum-containing DMM. What's shown on the x-axis is the seeding density the day before, and the images below show the percent confluency at time of transfection. Again, the titers are shown for the minus and plus enhancer component, and in each case, the enhancer boosts titers. Generally, we do not recommend that cells are overconfluent or covering the entire plate at the time of transfection. Also, we don't recommend that they're at a low density. as yields tend to be lower when there are less cells or when cell health decreases because of being overconfluent. So as you can see here, it appears that even if cells were accidentally seeded too high, use of the enhancer could counteract those negative effects on yield. All right, moving on from cell culture, which I just touched the tip of the iceberg on, using a shake flask or a bioreactor, different cell line, different media type, etc. These are all unique features of your cell culture system that can be considered when you're optimizing transfection. But DNA is just as important and unique. So not only the DNA quality, but the amount of DNA that's used, as well as the packaging and transfer plasmids that are used during transfection. So shown here in this experiment where we used VirusGEN LV kit with the enhancer with 500,000 cells per mil seeded the day before, we tested 2 DNA systems with different amounts of DNA per transfection. We maintained the same ratio of reagent at 3:1 micrograms of DNA. I should say 3 microliters per 1 microgram of DNA. And notice here on the y-axis, there is an almost 10-fold increase in lentivirus titer when the p-ALD system from Aldevron was used. And the optimal mass of DNA to use per transfection also varied. This really highlights that the DNA that you use is an important consideration for transfection optimization, not only for each unique cell culture system, but also for each unique vector that's being produced. In this experiment, I'm highlighting the effect of the ratio of reagent to DNA, so microliters of reagent to micrograms of DNA, as well as the amount of DNA that's used in the transfection. So these were AAV production experiments performed in suspension 293 cells with the enhancer in the VirusGEN AAV kit. In this experiment, we looked at 2 ratios, 2:1 or 3:1, and a 1 to 2 microgram of DNA transfected per milliliter of culture. And as you can see, the 2:1 ratio is clearly better for AAV titer than the 3:1, regardless of the amount of DNA that was used. For this particular experimental system and AAV vector, 2:1 at 1.5 or 2 micrograms produced the highest titers. So when you're considering doing an optimization experiment like this, you could test different ratios, like 1.5 to 1, for example, with a DNA titration. And GMP manufactured DNA, it can be an expensive resource. So optimizing DNA amounts used is definitely also important for this reason. Next, another interesting parameter to consider, which may not be on your radar, is complex formation conditions. And by complex formation, I mean the process of mixing DNA with transfection reagent. The resultant DNA reagent complex is what is added to cells. And so in this AAV production experiment, we examined the temperature of complex formation as well as the time we allowed complexes to form before we added them to cells. As you can see, high AAV titers can be obtained from complexes that were formed at either 4 degrees or room temperature. At 4 degrees, we think complexes grow a bit slower. For this experimental system and vector, we'd recommend an incubation time of about 30 minutes. And I also just want to make 2 notes that are applicable to all culture systems and transfection with TransIT-VirusGEN. And the first is that the TransIT-VirusGEN reagent should not sit in the complex formulation solution by itself for long periods of time. So we recommend generally that the DNA component is added first, followed by the reagent. If the reagent is added first, the DNA should follow within 5 minutes. And the second point that I want to make is that you do want to mix well after each addition. So after you add DNA, mix well. After you add reagent, mix well. But afterwards, we don't recommend additional mixing as additional agitation can disrupt the complex formation and formed complexes, which may have a negative effect on titers. And here is a similar complex formation time course experiment we performed for AAV production using the VirusGEN AAV kit with and without enhancer. And same idea here, about 30 minutes was optimal for yields for this experimental system. Playing around with other complex formation conditions can improve titers, and importantly, it should be considered for different culture and vector systems in order to achieve the highest titer. The last topic I wanted to touch on is the last stage of transfection. Not quite transfection itself, but the assessing of viral vector titer. And what I'm showing you here is a comparison of transfection for lentivirus production using the TransIT-VirusGEN reagent alone or PEI in adherent 293T17 cells. And this is data courtesy of Dr. Drobna, who produced lentivirus that had an RFP reporter. One of the vectors contained a control RFP in the A panels and another vector containing a Cas9 RFP fusion in the B panels. And these images show RFP transgene expression, most likely off the transfer plasmid that was used during transfection and not from the lentivirus that was being produced and secreted into the supernatant. These images were captured 17 to 22 hours post-transfection. Looks great that the transfection of the plasmids worked and VirusGEN is kicking butt over PEI. But transfection efficiency of the plasmids used to produce virus, it is important. However, I want to caution that using transfection efficiency is no substitute for titer. I think the data from this experiment I'm going to share in the next few slides really demonstrate that point. We transfected XP293 cells using the VirusGEN AAV kit with the enhancer component. We also included a negative untransfected condition, meaning no reagents or DNA were added to cells. And what we did was we transfected cells with plasmids that are required for AAV production. So in one case, we transfected all three of them. That's the transfer plasmid that encodes the GFP gene, the rep-cap plasmid, and the plasmid P helper, which contains genes required for high-titer AAV production. And in another transfection, we didn't include P helper. So in place of P helper, we used a non-coding filler plasmid. And when we evaluated transfection efficiency using GFP transgene expression, you can see that there are comparable GFP positive cells. That's what the bars represent. The dots represent GFP intensity. Even in the transfection where no P helper plasmid was included. So now I'm showing you the AAV titer, and you probably know where this is going. When we properly titered the AAV produced using a functional assay, you see that there is actually no virus detected when no P helper was included in the transfection. So if you were to just look at transfection efficiency or transgene expression post-transfection, you might incorrectly assume that there was an equivalent level of virus being produced between these two transfections. And so just briefly, how we did this functional titer was that we collected the sample after transfection, and we took that AAV sample and transduced or infected HT1080 cells with a limiting dilution of the virus. And from the number of HT1080 cells that express the GFP, we then were able to determine a functional titer. So I hope this experiment and the data here really makes the point clear that transfection efficiency should not be substituted for proper titering of virus. So what methods are there to properly titer or quantify how much virus is in a sample? And there are many methods, each with their pros and cons. You can check out this reference card from the Mirus website, and today I'm going to focus on these two in an example. We talked about functional titering being a measure of how infectious or capable the collected virus is at transducing a target cell. In the context of therapies, that would be how therapeutic is that virus sample. And another category of titering is known as physical titering, meaning not directly quantifying infectious potential, but rather a physical attribute of the virus. And this could be something like protein quantity of AAV capsid or lentivirus surface protein, or the amount of genetic material, DNA or RNA, packaged in the virus sample. Here's an example of functional and physical titers plotted side by side. For this experiment, we produced AAV using the VirusGEN AAV kit without or with the enhancer component, and we also transfected cells with these other commercially available reagents. The left graph here shows the functional titer that was done using that same HT1080 infectious assay that I mentioned earlier. And on the right are physical titers that were collected using a ddPCR-based assay to measure genome copies. As you can see, there are some differences, though the general trends in titer track between the two. And we also looked at harvesting AAV at 72 hours versus 48 hours post-transfection. So these are titers for the harvest at 72 hours. Here, side by side, I'm highlighting the AAV titers for the sample collected at an earlier time point, the 48-hour post-transfection. And as you can see, highest titers are achieved with the VirusGEN with the enhancer component, that is the whole VirusGEN AAV kit at an earlier time point, at least for this serotype culture system, and transfection conditions. And this is clearly reflected for the functional titer. And so based on this assessment for this system, we would recommend a harvest time of 48 rather than 72 hours post-transfection. Alright, so you probably heard me repeat the phrase several times today for this particular system, vector, and transfection conditions. So transfection is pretty straightforward, and the general protocol is the same. You add DNA, you add reagent to your cells. But to really eke out ever higher amounts of virus, you might want to consider optimizing transfection on a case-by-case basis by examining some of the parameters that I pointed out today. And the protocols that are provided by manufacturers are great starting points. So at Mirus, we are more than happy to work with you to discuss your unique workflows and systems and requirements for transfection. So to summarize, here are some potential variables you could consider for an optimization study. Obviously, one of the important outputs in such a study would be the viral titer, with the goal being to achieve maximal functional and physical titer. A true study of a matrix of all these variables is a huge undertaking, and here are some of the variables that I mentioned today. So going back to this image to remind you why you would even want to eke out more and more virus through transfection optimization. Clearly, quite a lot of virus needs to be produced in order to support gene and cell therapies today and in the future. So we can estimate the amount of virus that is going to be required per dose. So treatments, whether they're administered locally or systemic, will require a lot of virus. And if we take yield as these ballpark estimates here, you can see that the virus required per dose gets to be quite high in the hundreds to thousands of liter range in the case of a systemic treatment. And if you can achieve the same amount of virus with less cell culture and number of transfections, this would be extremely helpful, not only for material costs, but also labor and workflow costs, and especially precious time. So some concluding remarks here. High viral titers are required for these exciting therapies, and hopefully we're going to see more and more of these therapies doing amazing things in the years to come. And while transfection is just one aspect of advancing these therapies, I hope I've convinced you that optimization of transfection is a worthwhile endeavor to produce the highest titer of virus in order to support these gene and cell therapies. And lastly, we are happy that our TransIT-VirusGEN reagent alone, or together with the enhancer solutions in the VirusGEN AAV and LV transfection kits, can help in this journey to produce high titers of AAV and lentiviral vectors for these exciting therapies. If you are interested in testing the TransIT-VirusGEN reagent and or the VirusGEN AAV or LV kits, don't hesitate to reach out to either myself directly or through tech support by email or phone. As I mentioned earlier, Mirus scientists are happy to discuss your unique transfection workflows. And with that, I'll call it here and take any questions.
Thank you so much, Sandy, for that informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. It looks like, Sandy, we already have some great questions coming in from our audience members. Our first question, why are the ratios you start with are what they are? You use 1:1, but then recommend 1 to 1.5. Can you tell us a little more about that?
Yeah, sure. Happy to explain that. So I'm not quite sure where you saw the 1:1, but I think you're talking about the microliter of reagent to DNA ratio. And this is going to vary by what experimental system you're using and packaging mix, things like that. But essentially the general range that we came up with for our protocols is based on the amount of reagent that you can think of as a concentration that is provided. And so actually the ratio that we'd recommend working with first is closer to the range of 2:1 to 3:1. And anything that is dramatically different from this, like maybe something crazy like 10:1, would cause the complexes to form a little bit too quickly. Something that's dramatically smaller than this, let's say like 0.25:1, would have the reverse problem. So you can definitely get away with differing in the ratio there, but I would stick to the general range of 1:1 to about maybe even 5:1. Yes, I hope that answered your question. If you want to clarify more, you can feel free to add another question.
Thank you so much, Sandy. Now, what is your complex formation volume relative to your total culture volume?
Yeah, so I didn't think I talked about that, but that is definitely something that can be optimized as well. So to repeat, that's complex formation volume. That's essentially the volume of the solution that you use to form your complexes, where you add your DNA and the reagent to. And in our protocols, it's generally around 10% of the total culture volume. So if your culture volume is 25 milliliters, 10% of that would be 2.5 milliliters. And we've done some experiments where we drop that down to 5% rather than 10%. And what we find is the complex size grows a bit quicker. So if you do want to play around with your complex formation volume, you might also want to think about how long you're allowing those complexes to form.
Thank you so much. And again, I want to thank our audience members for these great questions coming in. Our next question, our validation process requires that we evaluate multiple lots of reagent. Is this something you would support, Sandy?
Yeah, so that would depend on, so we have different formats of our reagents. They're the same formulation, but we have an RUO, Select, and GMP manufactured version. And for the RUO and Select version, we already have several different lots available, so you could evaluate the different lots of those. The GMP reagent, which is going to be released later this year, about end of May, beginning of June, there is just one initial lot. However, following the initial release, I think in a few months following, we'll have an additional 2 batches. So depending on which format you want to evaluate, yes, there will be multiple lots to test.
Thank you so much. We have time for one more question. Can you explain your process for functionally titering virus?
Yeah, sure. So I kind of went over that in the presentation already. I mentioned that for the AAV2 that we've been producing the most in-house here at Mirus, we've been using HT1080 cells. And a functional titering method essentially is trying to measure how much of the sample can infect a target cell. So for the AAV2, because HT1080s can be infected by them, we use HT1080s. And what you do is you can count how many HT1080s are in a given well. You can apply your virus directly to the well. And what you're trying to measure is how much of those cells was infected. And those are called transduction units. And the way that you can count the number of cells that were infected, what we do is a flow-based or flow cytometry-based assay. So if there's a GFP reporter gene packaged in your AAV, for instance, or lentivirus, you can count how many of those cells are GFP positive using flow cytometry, and from there you can relate that to the number of viral particles that were in your sample that are functional. And since you know the volume that you applied to the well, you can get a concentration, transduction units per milliliter of sample. So you can design your assay based on what's packaged in your virus. So if you don't have a reporter gene, instead, if you want to use flow cytometry, you can use an antibody that binds your protein or gene that's being expressed in your virus.
Thank you so much, Sandy. And we actually have time for one more. I saw another one come in. How do you plan to use this study in the world? Like, what do you hope to accomplish?
Yeah, sure. That's a more, I guess, a more abstract kind of question, but I think it's easily answerable by the kinds of therapies that these viral vectors can support. So early in the talk, I mentioned these 22 that have already been approved worldwide that use virus. And so if you think about diseases where the treatment is some introduction of a gene or a correction of a gene, how do you introduce that gene, that corrective gene? The way that is safe and effective that we're finding is using these viral vectors. So they are being used now already and will be used increasingly over the years. And so what do you hope to accomplish with that? It's basically the treatment of these diseases that otherwise have no other treatments or cures. And in some cases, these viral vectors can correct permanently the disease, and rather than being a treatment, it can actually be a cure. So accomplishing just increased human health and happiness, I guess, is how these studies ultimately will help the world.
Sandy Tseng, thank you so much for your time today and for your important research. We really appreciate it. Before we go, I also want to thank our audience for joining us today and for their interesting questions. And I'd also like to thank our sponsor, Mirus, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast, by the way, can be viewed on demand for 2 years till April of 2023. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay safe and stay healthy, everyone. Take care. Bye-bye.